me share my screen. So yesterday we talked about guanides, glucosidase, and now we want to cover Ppar and a few of the other alternative and third and fourth line type 2 diabetes drugs. So remember, we want to be able to, when talking about type 2 diabetes, describe the role insulin plays in regulating blood glucose levels, describe the cellular pathways activated by insulin, and explain how can they be exploited pharmacologically, because remember, the different drugs act slightly different. Even the ones that are both targeting the ATP channel, right? They bind differently at different affinities. The potassium, ATP-sensitive potassium channel. Describe the mechanism of action of the major classes of drugs that are used to treat type 2 diabetes. For example, targeting of the ATP potassium channel. Understand some of the major side effects, e.g., be able to tell me what is hypoglycemia and how you treat it. We spent quite some time yesterday in talking about how to do that. And explain the rationale behind your choices for therapeutic options. So if I give you a patient that has a particular high BMI, and you should be able to tell me, okay, here's the test we want to run. Based on these tests, these are the treatments we're going to do, and this is how they work. All right, so we've covered majority of these. So now we're going to try and focus on, and we talked, so yesterday we covered the sulfonylureas, meglinotides, and biguanides. So today we'll cover those alpha glucosidase inhibitors and the more new drug treatments. And as well as pick it up with these glitazones. So what are these thiazolidiniones? The first version of these was this term glitazone, or troglitazone, and it was approved in the U.S. By the National Institutes of Diabetes. It's now removed from the market mainly due to hepatic failures. But there are newer versions of it that have been synthesized. One of those also off the market due to cardiovascular side effects. But there is still this one, pioglitazone, which is now the only one remaining of this family of compounds. And they act through binding of the peroxisome proliferator activating receptor, Ppar gamma. Ppars are involved in transcription of insulin-responsive genes and the regulation of the adipocyte lipid metabolism that we've talked about throughout. So how do they work? Well, in the presence of endogenous or exogenous insulin, glitazones serve to decrease the gluconeogenesis, glucose output, and triglyceride production within the liver. So the receptors are found on, again, our three favorite tissues when it comes to diabetes. Start modifying gene transcription within the adipose. This drives down lipolysis and drives down fatty acid, and then it's also going to decrease leptin and adiponectin. You're also going to see increased glucose uptake and increase in skeletal muscle within the muscle. And as I mentioned to you, the changes in glucose uptake and fatty acid use in the adipose. And Importantly, they also, these Ppar gamma inhibitors also can cause differentiation of adipocytes, which can be a strength depending on the type of adipocyte you're differentiating into. Importantly, because they work in different mechanisms from metformin, they can be used in conjunction with either metformin or the sulfonylureas, which makes them particularly attractive. Now, the Ppar gamma receptor has endogenous ligands made up of free fatty acids and iconosoids, which are small lipid-type particles. And these are found primarily in the adipose tissue, but also in muscle and liver. When the receptor is activated by these ligands, it forms a complex with retinoid X receptor and binds to the DNA affecting transcription of specific genes, which you'll recognize are involved in glucose and insulin response. So it upregulates Glp-4, the transporter for glucose. It modifies fatty acid transport protein, and it drives down lipoprotein lipase. And remember that important enzyme in metabolism, pepsi-K. So just a little video on Ppar modulators.
Activated receptors, or Ppars, are ligand-activated transcription factors and members of the nuclear hormone receptor superfamily. Other members of the family include the thyroid hormone receptor and the estrogen receptors. Like other nuclear hormone receptors, Ppars are ligand-activated transcription factors that control the expression of various target genes. Ppars form a heterodimeric partnership with an iron cis-retinoic acid receptor, or RSR. The site at which the Ppar ligand frequently interacts with the receptor is known as the ligand binding domain, or pocket. Any substance, including selected Ppar modulators, that binds to a Ppar to active receptor is called Ppar ligand. Activation occurs when the ligand binds to the Ppar RSR heterodyne, and there's
Lovely QQ, apologies.
After ligand binding, the Ppar undergoes conformational changes, which leads to the proactive. Association with proactive initiates the assembly of a large cofactor protein complex that carries out the transcription of mRNA.
So you can see the key aspect and key outcome for Ppar gamma inhibitors is that blocking of those key transcription factors, the transcription effect, and blocking of the gene expression. This pioglitazone should be taken, it's once daily, and the plasma levels should peak about 3 hours later of insulin. Plasma half-life is, the plasma level of the drug will peak about three hours later, and the plasma half-life of the compound is 3 to 7 hours. It is metabolized by the liver and excreted in the feces, and this important point is partly why there was some liver toxicity in that first generation of the compound. What are some adverse effects of these drugs? You get some dose-related weight gain, in part because of that adipose differentiation. You also get fluid retention, which promotes sodium-sensitive ion resorption in the renal collecting ducts, which can cause some edema, which will, of course, add to the weight gain. It is not known if it's safe for pregnant or lactating females. And it doesn't cause lactic acidosis, unlike some of the other compounds we've talked about, including metformin, even in patients with renal impairment. So that's one of the reasons it's still used today, is it can be useful for patients that are on renal treatment. And obviously, because of its role in the liver, it can start to damage the liver and get decreased liver function, which has to be monitored accordingly for the patients. It is subject to interactions, mainly due to its liver metabolism. So it can have, depending on what else the patient is taking, it can have side effects. And it can have the effect of reducing the ability of contraceptives to function, especially ones that are around estradiol or norethindrones. Okay, so that's the glitazones. Let's talk now about these alpha-glucosidase inhibitors. Again, these particular ones have an attraction that they are an alternative target and can be used in conjunction. So what are these alpha-glucosidase inhibitors? Well, they are they act on the gut and they're taken orally. They are basically delayed giant sugars, essentially, that prolong GI absorption of glucose, as these saccharides compete with carbohydrate digestion in the gut. Because they're so big and have so many binding sites that are similar to glucose, they can sit in the gut, and the gut becomes distracted, if you will, with trying to absorb these large polysaccharides, that the glucose sticks around and can still be effective. So here in this image here on the right, you can see these large polysaccharides competing with glucose in the microvillus, located within the gut. So this will lead to very small reductions in blood glucose. The nice thing about these compounds is they're not that effective, so they rarely cause hypoglycemia in monotherapy. They're often used to treat hypoglycemic patients. So you eat these monosaccharides along, such as with a glucose tablet. Importantly, these are, because of the way they function, they're competitive inhibitors. So it's important this is taken before meals. So you put it in before the glucose is in the gut. Now, obviously, because of the mechanism, major side effects are discomfort. So because you're putting in these large polysaccharides into the GI, you're going to get flatulence and diarrhea because the bacteria are also trying to digest these complex carbohydrates, and then will put off, obviously, quite a bit of gas. And again, because of the mechanism of action, in the function being mainly in the GI, there's really no major risk to hepatic toxicity. Now, just a reminder for what do we do with patients that present with hyperglycemia. We have a variety of options to us. We can add these or target this GI region, as I just mentioned to you. We can put in GLP-1 inhibitors and DPP-IV inhibitors that we'll talk about shortly. These are the new generation. We can add metformin or glitazone. we can, and glitazone that act on liver, or glitazone's also acting on the adipose and the muscle, or for metformin, it's mainly liver and muscle. So these are all the sites of action for trying to counteract hyperglycemia. Okay, so what about today? I've talked about drugs that have been developed over the last 50, 60 years. What about more modern drugs for treating type 2 diabetes? And if you look in the United States, they have a very funny habit. They love to advertise, and it's very strange because they always advertise medicines on the television. And these are this series of medicines I'm about to present to you are some of the most popular advertisements on U.S. television. It's very strange. Because here in the UK, they're not allowed to do that, or it's not very common. Okay, so let's talk about Glp-1 analogs. So just showing on the left here a incretin effect when you compare time and glucose levels with either given the patient is given oral glucose or IV glucose. Now, if you do the same experiment and you compare measuring insulin levels, shown here on the right, you can see a massive difference between whether or not the glucose is given orally or it's given IV, okay? Which suggests that there's something within the gut that is serving as a mechanism for modulating insulin levels, okay? And this got a lot of attention and became quite a heavy area of research to understand what the cells were, what the receptors were, and what were the molecules that were showing this differential effect in oral versus IV glucose. You can see this paper was published in the 80s. So it became discovered then that there was a peptide called GLP-1, or glucagon-like peptide 1, that is produced by the L cells, mainly located in the distal gut, ileum and colon, but secreted also from the proximal gut, along with a sister peptide called GIP, glucose-dependent insulinotropic polypeptide, which is produced by K cells in the proximal gut. Now, GLP-1 stimulates glucose-dependent insulin release, so that is partly for that effect in sensing glucose through the mouth, and this is mirrored in the K cells in the proximal gut by again GIP. In addition, GLP-1 suppresses hepatic glucose output by inhibiting glucagon secretion in a glucose-dependent manner, whereas GIP stimulates glucagon secretion. Okay, so here you've got counteracting peptides on glucagon. So immediately you recognize, ooh, these could be attractive drug targets if we can just figure out how to modulate them. And importantly, GLP-1 is an inhibitor of gastric emptying, which then will tell the body, okay, I'm full still, I don't need to eat, which will then lead to a reduction of food intake and body weight. And this last point was recently demonstrated in a very large international clinical trial. And so GLP-1 compounds are being taken forward for fighting and combating obesity. And if you remember, for type 2 diabetes, that is a major cause of onset of type 2 diabetes. So the GLP-1 target is particularly attractive for both obesity and type 2 diabetes. In the case of GIP, there's minimal effects on gastric emptying, and there's no particular effects seen on satiety or body weight. In addition to GLP-1, in addition to the emptying of the gastric, inhibiting gastric emptying, GLP-1 also appears to enhance beta cell proliferation and survival, which again really points to its attraction as a target for type 2 diabetes. GIP may also have this effect. So this just a slide highlighting the pleiotropic effects of GLP-1. There are also GLP-1 receptors in the brain, heart, and so one of the concerns, obviously, for targeting GLP-1 is on the cardiovascular side, but the attractive outcomes of liver glucose production, insulin sensitivity, and gastric emptying are a tremendous interest to the pharmaceutical industry. Because look, you can, seems like a wonder drug, right? You keep people from getting fat, you lead to better beta cell proliferation and increased insulin secretion and sensitivity, and you drive down glucose production from the liver. What's not to like, right? So it turns out that there are then a variety of ways to target this same pathway. First, initially, the compounds that were trying to target this pathway, because everyone thought peptides were difficult to make and to deliver into the body. So they first tried to target some of the processing peptides responsible for making a full-length GLP-1 or GIP in the body. So it turns out that in hypothalamus and in these L cells and K cells, you have some enzymes which are responsible for processing GLP-1 into a final, from pro-GLP-1, if you will, to a peptide cleavage activated GLP. So the idea was, okay, let's target these enzymes and see if we can't modulate GLP or GIP levels. So that's this DPP-IV that we'll talk about shortly. But then later in 2007, almost 20 years after the original discovery of the pathway, it was identified in this lizard. It's called a Gila monster, similar to a Komodo dragon, which is in Australia. They identified a very, very similar peptide, which is found in these animals, which serves to also act in a similar way to GLP-1. And then that told them, okay, if we extract this peptide, it's called exenatide, we can use it as a compound, as a drug, for treating humans. So that then told them that, okay, we don't need to focus so much on inhibitors, but we can also, we don't need to focus on the peptides, the peptidases, DPP-1 IV or 4. We can just figure out how to purify or synthesize this exenatide and use it in humans. So obviously, after a long amount of work, they were able to purify this exenatide, get its sequence, and then figure out how to use it in human. So they and make a synthetic version of it. Shown here is the excuse me, the amino acid sequence up above. And you can see this is that cleavage site of DPP4. But you can see that there are many, many conserved amino acids. So there's this human on the bottom and the glia on the top. You've got this nice WL, FI, alanine, everything spaced out very nicely that it could still bind to GLP-1 receptor. It surprisingly only has 50% identical sequence, which demonstrates the beauty of peptides and how you can modify them and still get activity. Attractively, it is also turns out that it's resistant to DPP4 inactivation. So DPP4 is responsible for cleavage of these amino acids to inactivate GLP-1. And so if you don't have this HGE, you will, if you have this HGE, and you don't have this HAE, you will not inactivate the peptide. It'll stay active in the blood longer. Now, these days, because of the diversity of peptide, there are other versions of it. The beauty is it's quite stable in the bloodstream. It still has to be injected just after or before the last meal of the day, but it has those wonderful pleiotropic effects where it can augment pancreas response, suppress pancreatic release of glucagon, slows down the gastric emptying, as I mentioned, reduces appetite, promotes satiety via the hypothalamic receptors, and reduces liver fat content. It is often used because it's a bit more expensive to make and for the patient, and it's a daily injection. It is often an adjuvant therapy for type 2 diabetics on metformin, sulfonylurea, thiolidinodiones. But for patients that are severely obese, it's more and more attractive and used. And importantly, obviously, because of the way it works, it is not for use in type 1 diabetes. As you can imagine, any side effects that are found are mainly GI tract in nature, including, obviously, stomach acid, belching, diarrhea, and heartburn. And there's been a lot of work on these side effects to try to reduce and make it more palatable as a medicine for patients to take. So how does it compare, this series of compounds, these GLP-1 mimetics, with endogenous GLP-1? Well, you still get increased glucose-dependent insulin secretion. You still get decreased glucagon secretion and hepatic glucose output. It still is able to provide that full feeling by reducing gastric emptying, reduces food intake, and it decreases plasma glucose acutely to near normal levels. And importantly, it's resistant to that DPP-IV degradation. And because of that, it is relatively long half-life within the bloodstream. Now, despite the fact that these are wonderful, there are other areas within the pathway that are attractive to target that we will go over, that in some ways were targeted previously to these GLP-1 peptides. And that is mainly because this last effect. They have high plasma concentrations and they're only injectable. And they have very strong effects on the receptor. So the idea was, okay, maybe we can make it modulate them and modulate the pathway in a different way and make them slightly less strong on the receptors. By instead of adding a mimetic, we can target those processing enzymes of GLP-1 to modulate endogenous GLP-1 levels. And that is the next family of receptors that are compounds that I will talk about. But first, let's take a little break, and then I'll finish it off with these DPP-4 inhibitors and the SLT inhibitors.